I'm often asked by friends and family, what am I doing in grad school? When I tell people that I study viruses, the reaction looks like this. When I tell them that I study archaeal viruses, the reaction looks more like this. They're wondering, what even are archaea and why study their viruses? Archaea are single-celled microorganisms, so originally we thought they were just bacteria. Actually, they represent a third domain of life, and they're more similar to eukaryotes. That's not the only thing making them unique. Many species of archaea exist in extreme environments like salt flats, hydrothermal vents, hot springs, and even inside cow rumen. They don't just survive, they thrive in these environments that would kill other organisms. So why study their viruses? Historically, the study of viruses has resulted in groundbreaking scientific discoveries about the species that they infect. Viruses have spent years and years of evolution figuring out exactly how to hijack their host cell machinery, and we can take advantage of this to study archaea by studying their viruses. I plan to do that to answer the question, how do archaeal viruses manipulate archaeal gene expression? To do this, I'm targeting archaeal virus proteins that are involved in manipulating gene transcription, also known as transcription factors. Unfortunately, the majority of archaeal virus proteins have no known homologs and their function is unknown. To overcome this, I've developed a computational pipeline based on protein structure to identify predicted transcription factors. Intro biology teaches us that when transcription factors bind DNA, they either block or recruit the RNA polymerase to start transcribing the DNA. When viral proteins bind viral DNA, this lets the virus transcribe its own genes. But when viral proteins bind host DNA, it regulates the host gene expression. My experiments show that my predicted transcription factors bind both viral and host DNA. So now I'm wondering, what happens when these viral proteins bind host DNA? To learn more, I've assembled a team that includes experts in virology and archaeal cell biology. Together, we'll use epigenomic and transcriptomic approaches to find out what sequences these proteins bind and how they affect archaeal gene expression. The funding provided by the Science Foundation would help me with sample prep and sequencing costs associated with performing cut-and-run and RNA-seq assays. If successful, we'll gain knowledge about archaeal processes for transcribing and translating genes. Currently, genetic tools in archaea are limited, especially when compared to bacterial and eukaryotic model organisms. However, because of their unique properties, archaea have great biotechnology potential. While this potential will likely remain unrealized for many years, fundamental studies like this are needed to set the foundation for future innovation.